Thank you, President Wang. So we have more time to hear from Professor Halldin's talk. We're very fortunate, okay? Because it's only 8:50 right now. We're 10 minutes earlier. So in the first session for this morning, we're very honored to have Professor Christopher Halldin from Karolinska Institute to share his insights in translational PET in neuroscience and drug development. Let us first welcome Professor Wensheng Huang from Zhengxin General Hospital to be our moderator of this session. Please, Professor Huang.
Thank you, the organization committee. It's my honor to introduce our first speaker, Professor Chris Halldin. Chris Halldin has completed his PhD program from Uppsala University in 1984 in Sweden, and then he go to a postdoc doctor scientist to be a postdoctor in Brookhaven National Lab, USA. And then he moved to his home country, Karolinska, since 1985, and be the assistant professor, associate professor, and director of Karolinska Institute PET Center in 2003. Now, he's the director of Karolinska Institute PET Center and professor of medicine radiochemistry at Karolinska Institute in Stockholm, Sweden. I think many of our colleagues in Taiwan has finished the PhD degree, including Professor Zhou and our previous radiologist head, Professor Guo, also from Karolinska. So many Thai connect of our colleague with Karolinska Institute. Halldin, Professor Halldin, everyone know him, so I not need further introduce of his history. We say more money, not more money. We say more time to Dr. Halldin to give us a talk. The topic is translational PET in neuroscience and drug development. Professor Halldin, please.
Thanks very much for this very nice invitation and the nice big present I got. Thank you very much. I would like to thank the inviters and the nuclear medicine group here in Taiwan, and I also like to thank my my previous students at Karolinska, Yuan Wanshu and David Young over here. Thank you. And I also like to thank Dorothy Yen. I don't know if she's here yet. And everyone else that I met during the years. I met some other people, Dr. Leo, Dr. Su, earlier this week. So old friends, it's very nice to come back. And my first visit here was about 25 years ago. And now I'm still here. Maybe my last visit, who knows? But still, I've been here many times, maybe 10 times. I've been here around, and it's always nice to see how as nuclear medicine have been developed very fast here in this country. So I'm always happy to come back here. So today I will speak about some principles behind how that we are working at the Karolinska Institute, and why we are using small animal, why we are using NHP, monkeys, non-human primate, and when we go to human and patient directly. So this sort of idea is behind my talk. So I will speak about a translational PET in neuroscience mainly, and in drug development together with the pharma industry. Okay, I will start here. So those of you that has been in Stockholm at Karolinska, now we have our new hospital. So it's a huge investment in Sweden, in Stockholm. It used to be 2,000 beds, now it's only 800 beds. So there have been some criticism, why doing this investment? But this is more sort of a treatment on special patient need sort of high quality sort of treatment, and therefore this hospital was built instead of the old hospital. And you can see behind here there is a second building, and that is our research building. So in the main building, there we have the nuclear medicine department. Maybe we have a PET CT camera, Siemens and GE camera. But if we like to do as research in patient, we have to do it in this building on the left side here, and here you can see it. It's called a bioclinicum. So it's 10 floors building, and under the basement we have U2 and U1. U2 is for the cyclotrons, and U1 under entrance level is for the radiochemistry. And then we have the imaging in animal and in human on other floors. So I will show you more in detail later on here. This is a radiopharmacy, U2, 2 cyclotrons, U1, many hot cells. It's 33 hot cells as lead shielded hoods. And if we continue, you can see all sort of new equipment. And we go on to level 4. Left side, we have the human imaging, all new cameras. And here is our PET CT from GE, PET MR, and other things. And in animals, you have all these instruments as well. And here is the 2 sort of newest camera. We have the PET CT, a whole body, but nowadays only 4 millimeter. We used to have HRRT, 1.5 millimeter, but nowadays you cannot buy that high resolution camera from Siemens or GE. So we have to buy this 4 millimeter resolution. And then we have a dedicated NHP, non-human primate. That is with at 1 millimeter resolution from Mediso in Hungary. So it's used for monkey only, but it can also be used for animals, small animals, rodent imaging. Continue here. So what we are doing at Karolinska is roughly, it's like 50% academic research, as you can see here, but also it's 50% with industry. So we do a lot for industry, for the pharma industry, because they want to develop new drugs more rapidly, and they want to find optimal dose of a drug without any side effect. So I can say that 80–90% of my budget is coming from contract with industry. So it's very important for us. I'll come back to that later on in my talk. So I think everyone know that we can use PET for tumor, heart studies, and brain, and mainly today in the clinic for Parkinson and Alzheimer's disease. You can see some examples here that you already know about. And FDG, of course, is the main tracer. Here is a tracer for Parkinson's disease. You can see it all the way from rodent, NHP monkeys, healthy control, and the patient, if you compare it with the healthy, is very useful. It's very useful tracer for Parkinson's disease. And I know it's already used here in Taiwan as well. Then we have one more example on Alzheimer. It's not C11PIB. It's a tracer that we developed with AstraZeneca. And you can see this comparison between the AD patient and the healthy volunteer very clearly. So it's a very low non-specific binding. So it gives the possibility to look on lower density and maybe earlier AD. Just an example of that. And so we are working in the translational approach, as you saw already, rodent, monkey, human. And this is the same tracer in all species here. It's a dopamine, a D2 antagonist, raclopride, that was developed already in the 80s at Karolinska and used all around the world. And you can see here the monkey as well. So I'd like to discuss about our strategy, how we are using different species in translational approach. We are also using in vitro studies, ex vivo, autoradiography. So we have a brain bank of human, NHP, and rodent slices of the brain that can be used. For example, here you can see one of them. And then we work in vivo, small animal, as many of you are doing, but we are only doing that in animal models, because we like to go into monkey as soon as possible, because the monkey in the primate is very similar and also predictive for human studies. That I will show you later on. In microdosing, when you are labeling the actual drug itself, just to see if it enters the brain, if it's meant for CNS. And we do occupancy studies you have to find the optimal dose of the drug with pharma industry. But we also, of course, they're using different tracers in as a diagnostic in different diseases. So now I'd like to explain when we are using small animal and when we are using monkey, before we go to man and patient. So why animal PET? Because it's due to the similarity of the DNA, and you can see that there is a quite good result in a rodent, but even better in monkey. And there are species difference between rodent and man. There can be sort of species difference in the PGP. They can enter the brain in different amount, and also metabolism is different. So we are using small animal for animal models, disease models, and we can also follow it for a long time in rodent. That's important. But of course, there are a different sort of advantage and different disadvantage with the different species. Okay, so here we have some example. Then in rodent, there are different animal models. Here is our scanners, very high resolution. I think there are already scanners from this company in Taiwan already existing. I think maybe there are 3-4 scanners in PET and in SPECT. So in Stockholm, we have the PET CT, is 2 of them, and 1 MR PET camera. And here is some example of that. FDG, there is a holder you can use for 4 animals at the same time. But there are also holders for 1 and 2 animals. So this can be very important. So just to show you, this is PET, CT, MR. So the CT is mainly for the bone uptake, as you know. And if you add the PET, you can see more of the biochemical information. And if you add MR, it's easier to see sort of the difference in gray and white matter. That are the principles of using sort of different cameras. So this is one example, dopamine D2 again, raclopride, and you can see injected and it's accumulated in the basal ganglia, in striatum, and you can fuse this PET with MR and get also the help of the anatomical sort of distribution. And this can be used in, for example, 1 animal model that I will show you here. In Huntington disease, we used 4 different tracers. Again, dopamine D2 in the wild type, and in the transgenic model for Huntington, but also we used PD10A, D1 receptor, and serotonin 2A. And you can see that it works more or less in all of these 4 targets. But we then went into patient, and we selected dopamine D2 and also PDE10A. And this shows then in patient. We have the D2 and the PDE10A in a stage 1 of Huntington disease, and there is about 75% sort of decreased uptake here in red of using PDE10A compared to healthy. But even in the pre-manifest, where there is no symptoms of this disease, you can use these tracers, even if it's a little bit lower signal compared to in stage 1. So it just is one example how we can work in the translational from small animal into patient. Okay, I continue. Why are we using NHP? I visited yesterday NMDC, yes, and they have monkeys nowadays. So why are we using that? Well, one reason is sort of the size of the brain is much bigger than in small animals, so it's easier to do, the ROIS, region of interest, compared to small animal. That's one of the reasons. And this is showing the map of the old site and our new site. And here we have our new imaging facility, and I call it Hilton Hotel. But now maybe I should change it then to Regent Hotel, because I'm staying in Regent Hotel, and that is maybe even better quality at Hilton. But why do we call it? And you can see here is an air shooter for the tracer. So it's like maybe 800 meters. So it takes like maybe 1 or 2 minutes just to transport this tracer underground from from this production to the Hilton Hotel. And why do I call it more Hilton Hotel? Because our monkeys are living in a very sort of natural behavior, and they can go in and out. They even have a pool. So I've been visiting in Singapore, other places, and I can see how the monkeys are living. But here, the only way for us to have monkeys is to follow the EU regulations, and we need to have it like this. And they are happy in this way. So then we are happy as well. So we have like 25 monkeys at the moment. They are under anesthesia, ketamine, ketamine to start with, and then under gas, sevoflurane. And this is again this camera I mentioned as dedicated for NHP. And so it's a full body, a whole body camera, a PET CT, and can be used also if needed for animals, small animals. But we are using this only for NHP. And all the monkeys are treated then like children. And all the journals and reports are needed in order to work with them in PET imaging. So what can we do in monkeys? We can use it for developing as new tracers, and we can use it for the pharma industry, as I mentioned, for developing of new drugs. And just 1 or 2 slides about chemistry. If you look on the short half-life, for example, of C11, if we do the chemistry and only look on the chemical yield, then we may do it up to 40 minutes, but we have this sort of 20 minutes half-life, so we should stop very early at 10 minutes. That is the ideal situation. So therefore, all chemistry has to be done very quickly. And we also need a cyclotron, and and we can use it for just to produce different isotopes, as you know. And in cancer oncology, F18, maybe copper, also gallium is used. In neuroscience, mainly F18 and C11. I think we are using like maybe 50-50 C11, F18 at Karolinska. And there are sort of different secondary precursors, as methylations can be done, or F18 using F18 minus. And if you look on the FDA, small molecules that are accepted by FDA, it's about 95%, they can be labeled with either F18 or C11. So many structures can be labeled if we are interested, for sure. And but just to find a like ideal tracer, then we need to fulfill like a list of important criteria. If like only one of them are not fulfilled, as this tracer will not work in PET. For example, it needs to enter the brain, not be a substrate then for PGP. It will not enter the brain. And no labeled metabolites, because on the PET image, if we have that entering the brain, it will be like a mixture of the unchanged parent and a metabolite that are labeled. Then we cannot use a tracer. And of course, it needs to be potent and selective for that specific receptor or target that we are interested in. So what is an ideal tracer? Here already in the 80s, of my colleague Lars Fade, raclopride, again, dopamine D2, it's a fast initial uptake and a fast washout. And here is the uptake in the reference region with low density of in this case, a dopamine D2 receptors. And if you take this one minus this one, this is a specific binding. So in principle, then we can stop at about 40 minutes, the scanning. So this is ideal. If every tracer looked like this, we would be happy. But they are not looking like this. This is very complicated, in fact. We have to follow this list, and we have to test it in monkey or in rodent. But if you have monkey, better to if you do it in monkey. And so our experience, even from 1982, is that we have injected up to 300 different ligands into monkey, but only 60 of those were that sort of promising that they could be injected into man. And only 25 of these 300 are useful. So if we didn't have the monkeys and would have been sort of going into man directly, there would be an 80% failure. Therefore, it's good to have the monkeys and do the experiment sort of initially in monkey before you go to man. So I got two examples. One is a Japanese company. So this is published, so I can show it. PDE10A. So what I did, I looked on this list of important criteria, and we selected seven different compounds. And we looked on the binding affinity, selectivity for the target, and log D as a lipophilicity, and if it's a substrate then for PGP or not. If it is, it will not enter the brain. And there is a threshold for that, for example, 1.8 and 2 are in the borderline that they may not enter the brain, but we need to still have to show it in monkey. So if you remember number 3, number 9, of this one you can see the uptake in brain, and number 3, number 9 are not taking up in the brain. So it's important to know this sort of binding characteristics before you start any study. But this is, of course, only images, and then number 5 and 6 looks maybe promising. And here are the numbers, uptake in brain. Blue is 5, red is 6. There is a high uptake of these two and a fast washout. And if you look on this a specific binding again, striatum minus cerebellum, we get this sort of specific binding, and number 6 is looking very promising if you compare with what I showed you earlier of the ideal tracer. And then number 6 was used in patient for the company. This one was used. So second example is a serotonin 1B tracer. So AstraZeneca had like 3,000 compounds in the storage. And I looked on this list again, and I selected 7 or 8 of those, all very easily then to label here with the N-methylation C11, and it looks like this in monkey. Even number 5 is not entering the brain. Even if the company thought this should enter the brain, it did not. So which one should we select here? We know that there is an uptake in the occipital cortex from in vitro autoradiography. So yeah, perhaps you would guess that number 2, number 4, number 6, number 8 are promising. And number 6 was selected due to a nice uptake, as I showed you earlier, and a fast washout, like this. Yeah, so, but afterwards, we were interested in a rodent animal model. And as we injected the same tracer that entered the brain of monkey and human, but it did not enter the brain of rodent, rat. Then we gave like an inhibitor of the PGP, and then it entered the brain. So my message is that if we would have been starting in rodent, maybe this tracer would have never been used in pet in man. So therefore, we have to be careful with rodent experiment. Better to go into monkey directly, at least in this case. Okay. Now I will go into my last and second part of my talk, as using PET for industry. Why are they so interested using PET? They are interested in both in occupancy studies, and to find the optimal dose of a drug with no side effect. And then that we call also as target engagement. And then we also like to know the uptake in brain as brain perfusion. Is this drug entering the brain or not? So occupancy studies is used then for dose finding. Here is one example in monkey again. if we start in monkey, baseline after 2 hours, before the second injection, we inject this concentration of the drug, and you see there is the occupancy of lower uptake in the occipital cortex of this tracer. And in the third experiment, same day, after 2 hours more, we give 8 times a higher concentration of the blocker, and you can see even higher occupancy. So it's very easy to do this before you go into man. So what I just showed is we have a baseline and 2 different doses. You get maybe this occupancy, maybe 10%, and you get maybe 85, 90%. And then we just continue in a second monkey, baseline and different doses like this. And in the last one, in the third monkey, sort of the same paradigm. And then we can plot this occupancy then to the plasma concentration that we need to analyze. So this has been used by at Karolinska already early 90s for antipsychotic drugs that is used for schizophrenia. And you can see that it should be like 70 to 80% occupancy. And if it's higher, then you have the side effects, extrapyramidal side effect. And under 70, you may not have any effect of the drug. So that has been used at Karolinska by Lars Fare. In blue, you have the EPS effect, and 70 to 80 here is what is recommendable. For classical antipsychotics. So just a way to show what every company nowadays wants to do. Occupancy study, you have to find the ideal dose of a drug with no side effect before they go to phase 3 and phase 4. Okay, and my last, again, you can see here, is really monkey occupancy study, is it useful before you go to man or not? Yeah, you can see here it's almost identical KI value, so it's predictive for human. So the company still wants to do it in man, even if we have a data result in monkey. And this is the same tracer that I showed you that was not entering into rodent brain, but is entering into monkey and into man, like this. Okay, so my last part of microdosing, what is that? Just to show that a labeled drug is entering the brain. So we don't have to, use a tracer for PET, that is ideal. If we can label the drug itself, as I showed you earlier for FDA, almost all of the drugs, they can be labeled with C11 or F18. So if we are labeling a drug itself, and we show that is entering the brain. So because if there is no uptake of the drug, it is one of the reasons for failure of drugs in CNS. You can see here in monkey, here's the brain, here is the mouse. So this one is taking up in the brain, monkey. This one is not taking up in monkey. And then again, industry like to show it in man, and this one was also a failure. It did not enter into brain, and it was stopped early on. So it's good for the company, they save money, and it's good that we don't have to inject it into a large number of subjects or use it as a drug in phase 3 or phase 4. Okay, so I think this is just a list of all companies that we are working with. And just like a summary, since 1985, we have like 0.8 billion Swedish crowns. This must be like 80 million US dollar or 80 million euro as a profit from our contract with industry. So as I said earlier, 80–90% of my budget, for 50 people, and all the scanners that I showed you is coming from interaction with industry. Even if we also have, of course, grant with as Michael J. Fox and others, industry for me is very important. And we have published since 85 more than 700 articles, and if you look on those 700, it's about 300 that are published together with industry. So they are co-authors on these papers. So it's not only the funding, it's not only that I'm getting new compounds and new targets for research. It's also that we can publish these articles. And it's 37 different theses from 1985, and there are 2 of them are here today, Johanwa and David. And so it's really important for us. And just like a joke at the end, but maybe it's not a joke, that without imaging, maybe we are coming sooner to heaven. Early detection is important. And last one is my group, and I think we can see David over here, but not Yuanhua at that time. You have already went back to Taiwan. Okay, thanks a lot.
Okay, very thanks, Professor Halldin's excellent talk. From his talk, we know that his institute is very unique and full equipment and full manpower, and do top things in the world. So I think, Aina, you should learn from Karolinska. This is your opportunity. We still have 8 minutes. We open several questions to the audience. Is there any comment or question from our audience? Okay, Professor Lee, yeah. From Cancer Hospital.
Hello. Nice to meet you again. I saw you this morning and talk short in the elevator. So it's my honor to hear your talk again. As your talk, you say that your funding are more than 85% come from the industrial funding. Yeah. In my opinion, maybe it's my question. It's a good phenomenon for the academic research, or is it good or better or not?
So did you ask if it's good that I have so high number, 85% or 90%?
No, because industrial
from industry.
because the industrial funding come from the big pharma, they own their direction of the new drug direction.
Well,
But academic research is a new idea or a random idea come from the
I
somebody.
think it's a very good question, but me and my colleague, we have been thinking about this over the years, and industry are working on new targets, for example, new targets, and they need also PET tracers for the new target, and they come to us with a new target that I didn't think about. So I can work on new targets with industry, and they have all the compounds, 3,000 compounds. So for me, it's looking very good, but it's more simple for me to work with industry. They have the targets, they have the compounds, and I only have to label these compounds. Yeah, with my skilled chemist. And in addition, they also pay for it. So is it good or bad? Of course, there is some limit that maybe my group of 50 people would not sort of develop their own ideas, perhaps. But after all, as we can publish many articles and many PhD, I showed you that. So I think, in summary, it's good for me. But we have to be thinking maybe twice before we accept like a new product. If that was your question, I'm not sure. It was? Okay.
杨先，
请问还有没有各位有没有什么这个问题？特别是在研究机构啊，发展那个药厂。
Thank you, Professor Halldin, for your talk. Are there any other targets other than brain for the microdosing therapy? For labeling, other than brain, any like heart or other target organs that are in the near future, or
definitely yes. So the major pharma has sort of moved from the CNS. AstraZeneca, for example. We are still working with Astra very heavily, but in the whole body and cancer and oncology. Now I only showed what is published things, but yes, we are working with industry in oncology and cancer also. But so if you look on the radiochemistry, that can be used for both CNS and for whole body and cancer tracers. Maybe as different isotopes, perhaps. Maybe not C11 anymore, but that can be gallium-68, zirconium, copper. Also peptides can be labeled. So yes, we do that also in other than the CNS nowadays.
Thank you very much.
Professor Halldin, your microdosing radioligands are identical to the drugs provided by pharmaceutical company? The radioligand you use is identical? The drug?
Okay, so your question was that can you have like an ideal tracer of a drug?
Yeah.
Yeah. No, usually like a good drug is not a good tracer. For example, I showed you this 3,000 compounds. Maybe one of them was selected as a drug, but it's not useful for PET tracer. And because binding affinity has to be high for PET tracer, and maybe for a drug, it doesn't have to be very high. It needs to be showing as a target engagement for the target, but an ideal tracer is not the same as a drug.
Thank you. 还有2分钟，
Yep.
对那边。From Taiwan University.
Yes, Professor Halldin, I'm Dr. Chen from, we met last Sunday night, if you remember.
So maybe yesterday we met.
No, it was the last Sunday night, but that was okay. I was just wondering, is there any newer tracer development for imaging the peripheral nerves? Because we have a lot of patients with peripheral neuropathy, and our clinicians are very, they want to know the degree of the neuropathy and how we can use the different drugs to treat them. So because now we have a lot of tracers for the CNS neurological system, but we have few for the peripheral neuropathies.
Can you summarize?
Do you develop radioligand for detecting peripheral neuropathy?
Peripheral uptake?
Yes.
I mean, when we do it? And
To detect peripheral neuropathy. Because most of our radioligand is focused on the central, not peripheral.
yeah.
She asked, is there any radioligand for detecting peripheral neuropathy?
Yes, it is. And I think the same question as you had earlier, that you can also work outside the brain, and especially in cancer oncology. Also, there are tumors in the brain, of course. So there can be tumors in the brain and outside the brain. Sometimes we have to select different traces for brain and outside the brain.
Right, but because the peripheral neuropathy, the axons and the fibers are very delicate. They are very small, so it's tracer used for the CNS will be a little bit difficult for imaging the peripheral nerves.
Yeah, I think you are right. There are different sort of questions outside the brain and brain that you have to answer. Now I spoke only about the brain, but outside the brain cancer, theranostics is a new area where you have both drug and a labeled diagnostics, theranostics. So that is very popular today. Many centers are starting up with theranostics.
Okay.
So just to answer you very quickly, it's different questions maybe outside the brain and the brain. And as metabolites, for example,
Right.
I show that as labeled metabolites should not enter the brain. But in the full body, they are maybe everywhere.
Okay, thank you.
So you have to know which metabolite and where are they accumulated in the whole body. Sort of questions like that, yeah.
Okay, thank you.
Yeah.
I think that the time is due, so we make the accurate end of this talk. Thank you, Professor Halldin. Excellent talk.
Thank you, Professor Halldin and Professor Huang. On behalf of the Taiwan Society of Nuclear Medicine, we invite Professor Huang to give Professor Halldin a gift for the inspiring speech. Thank you.